Thank you, Kurt. That was a wonderful introduction. It's my co-supervisor, and I was supervised as well by Alan Ellis. So let's jump into integrative approach to species delimitation of Corsomyza. So when we talk about species, we talk about how they are foundational units in biology and used as a metric unit in biodiversity studies and conservation. And understanding what a species is important for conservation works in areas with high densities of species, such as biodiversity hotspots that can only be identified when we are able to accurately delimit species. Misidentifying species can lead to poor decisions about where to direct conservation resources, so understanding and classifying species correctly isn't just academic, it's essential for protecting the world's biodiversity. And when we ask this question, what is a species, this is a very normal response, because it's often difficult to vocalize how we determine what a species is and how to separate them. To address this philosophical question of what a species is, there have been numerous species concepts outlined to push us forward towards an answer, and defining a species is usually determined by a species concept one employs. While there are numerous concepts, these three on the board are the most commonly used. And while each of these has a role to play in delimiting species, there are surface level classifications that can often have pitfalls that do not adequately address species boundaries. For example, the morphological species concept does use diagnostic characters to determine species boundaries, but it merely infers reproductive isolation depending on genitalia morphology rather than ensuring it. And when we use single species concepts, we investigate species boundaries at a surface level. However, if we marry species together, we can investigate boundaries at a deeper level. With groups that are well-resolved taxonomy and active taxonomic experts, species delimitation is historically estimated on morphological distinctiveness, and then the hypothesis is retested using phylogenetic analyses. And phylogenetic investigations can help to identify synapomorphies. In groups with taxonomic impediment or the lack of taxonomic expertise, we essentially have to start again, where we first assess the reciprocal monophyly of groups to identify genotypic clusters, and the basis of each group that is monophyletic is a species, and phylogenetic analyses is instrumental in pairing males and female representatives that may be absent in current taxonomic tools. After molecular species boundaries have been drawn, we can then use distinct morphology to identify species in situ. However, a caveat is that molecular analysis doesn't always mean that the morphology will be identified. So let's shift gears to South Africa, and particularly the Greater Cape Floristic region. The GFCR has incredible floral diversity, and this is attributed to the unique landscape that make up and abiotic factors that floral lineages experience, and that stimulates divergence. And the GFCR does harbor high insect diversity. However, levels of endemism and species richness are unknown for most insect groups. This knowledge gap is attributed to the lack of capacity to tackle baseline diversity studies, outdated or absent taxonomy, and the amount of effort it would need to take just to do these kind of studies. So essentially, the insect taxonomic effort and expertise in the Greater Cape Floristic region lags behind that for plants. And why should we care about insect diversity? What is the relevance to the flora of the region? Well, during spring, there's mass blooming events here with predominantly daisies. And when we look at the pollinator abundance, we can see that flies and beetles are the most common pollinators of daisies in the Greater Cape Floristic region. And it's quite interesting to note that there's a shift in pollinator abundance between two regions, Namaqualand and the Western Cape. And these differences in pollinator abundance can occur in the same species in each region. For example, Dimorphica cuneata, where we have shifts in pollinator abundance.
Yeah.
And a large portion of the fly pollinators we see belong to the subfamily Maribizinae. While there have been numerous studies on the monotypic genus Megapulpus and that suggest it's a keystone species in the Greater Cape Floristic region, there have been no investigations primarily focused on the pollination ecology of other genera. And this is largely due to the taxonomic impediment of the group. Now, if you ask a taxonomist to identify Corsomyza to species level, they will audibly groan. It is hard and it's not fun. And there are a few reasons for this difficulty. First, if we look at the timeline for Corsomyza taxonomy, Corsomyza was described in 1820 and not placed in any tribe or higher level taxonomy order. So there's no further hypothesis surrounding Corsomyza and its related genera. More than 100 years later, and Corsomyza were placed with other genera in the subfamily Bombyliidae by Hess, and this literature is still the recommended species key for Corsomyza and most of Maribesine's genera. Since then, Corsomyza has been placed in the subfamily Maribesineae, and the subfamily's relation to other subfamilies has been investigated further. But there's still some work that needs to be done. While there has been some work done on the other genera from Hess's key, it's still a recommended tool for species identification, and once we look at the key itself, some other issues come to light. So the first problem with Hess's key is that he described a large portion of Corsomyza, and he has been critiqued for oversplitting species based on characters that you might consider intraspecific variation rather than species-specific traits. And most species that were described were single representatives or from a single sex. And this means that intraspecific variation is not well addressed in the current taxonomy. So to address this oversight, here's just an outline of what I've done so far. Well, what I did. Due to the limited number of Corsomyza specimens in the collections, the vague nature of type localities, and a lack of understanding of intraspecific morphology, I adopted a systematic widespread sampling strategy across the Greater Cape Floristic region in South Africa. At 93 of 170 sites across this region, 652 Corsomyza specimens were captured. Specimens were either dry preserved or we preserved them in ethanol for later molecular analyses, and legs were stored for generating a barcoding database. Once I was back at Stellenbosch, I tentatively identified specimens using the current taxonomic tools, and since either the male or female of most described species is unknown, I tentatively assigned names to specimens that did not match described species, but were grouped in the CO1 tree with known described species of the opposite sex. 2 collective morphospecies, as I have photographed here, with unique paddle-like antennae, did not match any species description and were treated as undescribed. So for the first step in what I've done, I generated a barcode gene tree, and from there I split the tree into what are called major clades, and this formed the minimum species diversity hypothesis for this genus. From there, I identified the subclades within each major clade, and took on my molecular diversity from 6 to 18 possible species. Next, I assessed the molecular diversity using molecular species delimitation models, 2 which use the Barker gap to infer diversity, and 2 that use tree topology. While 3 of the models aligned closely with Hess's initial estimates of species diversity, the one just overestimated diversity completely. After this, I investigated the morphological diversity within each major clade to determine whether morphology of specimens aligned with the molecular diversity estimates. I'll use clade B as an example for how this looked. So to explore the morphological diversity, I used a binary matrix to score the presence or absence of morphological traits and used a principal coordinate analysis to plot clustering of individuals. From this example, we can see that clustering of individuals closely aligned with the identified subclades, and these subclades have single morphologically distinct species. Post hoc observations of these putative species allowed me to pair what I hypothesize as a species with the current type specimens. So all this work can be summarized with the following table. From the Bayesian inference CO1 tree, there are 6 major clades that were the minimum species diversity for this genus. In each major clade, there were a number of subclades, and when I investigated the morphology for each subclade within each clade, I saw distinct clustering and was able to identify diagnostic characters that can be used to identify putative species based on these results, and I could then also link it to the current type specimens. And the results from what I've done show that species diversity within Corsomyza has been greatly overestimated. I recovered 18 species represented by the subclades within 7 major clades, while barcode delimitation approaches suggested 21 to 21 molecular species. These diversity estimates are largely incongruent. However, when integrating the morphology with the molecular, I saw that there was support for the molecular delimited species based on the subclades. I found support for 16 putative species, and that included 2 previously undescribed species. This is substantially lower than the current 27 recognized species within Corsomyza, and really just suggests it's been overestimated. While you could say that the lower estimated species diversity is due to not all specimens being collected, the wide sampling really did show that we've collected numerous species, and I reject the idea that we just didn't catch everything. This has all really helped to resolve the taxonomic impediment within this group, as we've been able to pair males and females, as well as capture the wide interspecific variation within Corsomyza. The only caveat within this investigation was the use of the single gene model. The CO1 gene region is affected by geographic separation, so specimens that might be collected at the same site would be seen as more related than those that are further apart. So if anyone wants to use this type of methodology further, I really recommend using a nuclear region. All right, and I'd like to thank everyone who was involved in this project. It was really great.
Okay, thank you very much. Well, in time, we have time for a few questions. She's a postdoc now, so she can have something. Yeah, so. No one? Then indeed, it was a very clear presentation, so we wait for another 3 minutes for our next speaker. Thank you.
Yeah.